Hi students, today we are going to discuss about what are all the outlines or the important points for beta oxidation assay question. Already I have uploaded the pathway cycle related points. Here I am going to discuss about the what are all the important outline or points for your essay question, because this is the number one repeatedly asking essay question from lipid metabolism. That is why I wanted to put one more video for the outline. With this outline only it will be useful for your essay question, right? As usual, first point is you have to define what do you mean by beta oxidation, right? Successive and continuous removal of two carbon atoms from one long chain fatty acids at beta position, right? Second one is what is the site and location. That means which organs or which compartments, both you have to say. Here the organ is liver, muscle, heart and kidney, some percentage of kidney. Site is usually the first step occurs in cytosol, the remaining reactions occurs in mitochondria. So both cytosol and mitochondria is the answer for your site for beta oxidation. Very, very important MCQ point. Next one is what is CoA? For beta oxidation, don't forget to mention about CoA, that is coenzyme. This is very, very important for your first and last step of beta oxidation, right? Next one is what is carnitine? Actually, we have three types of fatty acids: short chain, medium chain and long chain fatty acids. Short chain and medium chain, that is less than 12 carbons, they will enter freely into the membrane. Whereas more than 14 carbon atoms, that is long chain fatty acids, the membrane is not permeable to enters into the mitochondria from cytosol. That is why we need carnitine, which is the transporter for the transportation of from cytosol to mitochondria, right? So what is the composition of carnitine? What is the chemical name? Amino acids, synthesized from which amino acids, from which organs, what is the role of carnitine, what happens in carnitine disease, like that you have to read. We have uploaded already separate video for carnitine itself, so at your free time all of you go through, right? Second one is, this is carnitine is for what? Transportation of fatty acid from the cytosol to mitochondria, right, in beta oxidation. Next one is first step takes place in cytosol, that is conversion of fatty acid into acyl CoA, right? Very, very important points. Conversion of fatty acid into acyl CoA in cytosol. Next four steps, they are taking place in mitochondria. Through the carnitine, the acyl CoA enters into the mitochondria, where the further four oxidation reaction, exclusively oxidation reaction, takes place in mitochondrial matrix. In these four steps, two dehydrogenous steps are there, very, very important steps. That is ATP production from NADH and also FADH2 occurs in these four oxidation reactions. And the last step is, another one is hydrolase, that is hydration step. That is not significant, but last step is very, very important. The enzyme name is thiolase. With thiolase, the end products of this beta oxidation is one molecule of acyl CoA and another molecule of acetyl CoA, right? Then what is the fate of this acyl CoA and acetyl CoA? Acyl CoA again come to the first dehydrogenation step for the next cycle of beta oxidation, whereas acetyl CoA enters into electron transport chain for production of ATP. Actually, you have to draw one diagram for one cycle only. In the last step, you have to mention 7 plus 7 cycles or totally 8 cycles, like that you have to mark. That is why diagram for one cycle, and you have to mention in the last step totally 8 cycles or remaining 7 cycles, like that. Because in beta oxidation, one long chain fatty acids undergo totally 8 steps, right? So coming to the regulation, how it will get controlled. We have 4 points for the regulation. Very, very, very important, because sometimes Regulation of fatty beta oxidation also your 5-mark question. First one is availability of the substrate. That means fatty acids when you will get after lipolysis only. If lipogenesis occurs means there will be no place for lipolysis. So lipolysis genesis decrease means lipolysis will get automatically started. So availability of the substrates, that is fatty acids from lipolysis. Second one is the exclusive pathway enzyme from the pathway is thiokinase. If the fatty acid synthesis enzyme activated means beta oxidation enzyme will be subsided. That is the importance of thiokinase. Here I am mentioning two compounds, that is CAT1, carnitine acyl transferase 1 and melanyl-CoA. Actually melanyl-CoA is the regulatory enzyme for fatty acid synthesis, that is lipogenesis. If it is activated means this carnitine acyl transferase 1 will get inhibited, right? If it is beta oxidation enzyme CAT1 is stimulated means automatically melanyl-CoA is decreased. Last one is hormonal regulation, very very important, insulin and glucagon. Here the important MCQ is glucagon is stimulated beta oxidation, whereas insulin is inhibited beta oxidation, very very important point. The concept is glucagon insulin ratio is more in beta oxidation, whereas in fatty acid synthesis or lipogenesis the insulin glucagon ratio will be more. This is very very important MCQ point. Next one is from this pathway how many ATPs you will be getting, that is acetyl-CoA 8, NADH 7, FADH2 7. That is 8 molecules, 7 molecules, 7 molecules. On the whole, totally you are getting 108 ATPs. Actually, for the first cycle, we are giving 2 ATPs for the activation of the fatty acids. So you have to minus 2. That is 108 minus 2, totally 106 ATPs from beta oxidation. Actually, for this ATP production or energetics is your again 5-mark short notes. We have already uploaded separate video for this energetics calculation. At your free time, you go through please. So next one is what are all the disorders you will get from this pathway? We have 2 disorders. One is regarding carnitine deficiency. This carnitine deficiency is very very important because this deficiency is having 2 important symptoms. One is hypoglycemia, another one is muscle weakness. That is why carnitine deficiency is very very important. Question mostly MCQ question. Next one is organic aciduria. This is again 5-mark short notes. I have uploaded separate video for organic aciduria. So at your free time you go through. And final point is for this essay question you have to draw 3 separate diagrams, or on the whole you have to club all the 3 in one pathway, that is activation of fatty acid, second one is carnitine, third one is beta oxidation, specific oxidation reactions, right?